Dear viewers, this video is part 4 in the 4-part series on infections overview in Medicine for Anyone in the YouTube channel. The immune system. The survival of pathogens in hosts is made particularly challenging by the existence of the immune system. Pathogens have responded to this challenge by evolving many specific mechanisms for the avoidance, evasion, or subversion of both innate and acquired host resistance mechanisms. High mutation rate. For example, some viruses are inherently genetically unstable. The polymerases that replicate the RNA genomes of influenza virus and lentiviruses such as HIV result in variants being produced at a high rate. Consequently, many defective viruses can be detected, although they are usually missed by conventional sequencing approaches. Genetic changes may confer fitness benefits, leading to selection of the variant viruses. In this way, over time, progressively varying viruses evade the immunity that develops in response to infection. Gene swapping by viruses. Some viruses, such as influenza, have evolved segmented genomes. If two viruses happen to be occupying the same host cell, different segments can rearrange like shuffling a pack of cards and a new virus can be assembled. Antigenic variation when inside the host. At an entirely different level, within host antigenic variation is thought to be one mechanism that allows HIV to continue to be carried chronically in the face of what might appear to be a strong immune response. Bacteria such as Haemophilus influenzae, Neisseria meningitidis, and Campylobacter jejuni have evolved tracts of repetitive DNA in a single base pair repeats. The number of these repeats can change apparently randomly. This is a powerful mechanism that allows the existence of a population of bacteria with several different antigens that may be randomly expressed or not expressed in individuals within the population. Thus, some members of the population are lacking a particular structure which is itself a target for the immune system, such that they will survive and thus continue the existence of that bacterial population. Mimicking innate immune response. Pathogens have evolved mechanisms to subvert the immune system by mimicking elements of the innate response. Good examples are herpes and pox viruses that encode chemokine homologs and or chemokine receptor homologs or analogs. So, dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Genomes. Many bacterial, viral, fungal, and protozoal genome sequences are now complete. Now, the real challenge for researchers is to sift and unravel the whole mass of information and to select from it that which is genuinely useful. The future prospects. Most infectious diseases are unlikely to be eradicated in the near future. Even if they were, new infectious agents would inevitably evolve to exploit the rich environments presented by host animals, including humans. The infectious diseases have the capacity to deliver sudden, severe global pandemics, resulting in high global mortality. Emerging infections presenting acute public health problems are likely to be viral in nature, to have originated in an animal population, and therefore, at least initially, to be zoonoses and to be spread quickly via air travel. Changing social conditions, example, increasing urbanization in certain parts of Africa, bring together animal and human populations that have rarely, if ever, been closely associated on a larger scale. This brings with it an increased chance for microorganisms to be shared between these species, with a resulting increased chance of new pathogens emerging. The devastating effects of Ebola in West Africa from 2013 to 2016 exemplify this perfectly. Even relatively minor changes in societal behavior can lead to major disease problems. For example, changes to methods for preparing cattle feed led to the emergence of the bovine spongiform encephalopathy (BSE prion) as a human disease problem in recent years in the United Kingdom and elsewhere. Explanatory note: the BSE epidemic resulted from the feeding of cattle with mammalian meat and bone meal derived from cattle infected with BSE. Experiments have shown that as little as 1,000th of a gram of BSE infected grain material is sufficient to infect a calf. Effective feed controls are a key to the eradication of BSE. Most pathogens infect more than one species of host animal. A concerted effort is needed to deal with pathogens in all parts of the world and not just in the developed world. The lives affected by pathogenic microorganisms in developing countries are just as valuable as those in more affluent areas, but are far more numerous. A focus on primary medical care and early detection of outbreaks would be far more beneficial than high-tech approaches that some claim will enable us to predict which specific pathogens will cause future human pandemics. Infectious diseases of humans involve intricate interactions among the infecting microbe, human tissue, and the host microbiome. Infection process. The process of infection can be divided into several stages. The encounter with and entry of the microbe into the human body, that is colonization, the attachment of the microbe in its favored niche, and microbial avoidance of host defenses, that is infection, the combined deployment of microbial factors that damage human tissue and host inflammatory responses to the presence of the microbe, that is disease, and the release of the pathogen into the environment where it can infect others, that is transmission. It is notable that for most microbial pathogens, the host inflammatory response contributes substantially to symptoms and to tissue damage. Moreover, the human microbiota, that is the collection of microbes that reside in and on the body, modulates directly or indirectly every stage of infection. This discussion describes the molecular and cellular mechanisms that contribute to human disease caused by bacterial pathogens. Entry into the human host. Infectious diseases occur when either a live pathogen enters the human host or a toxic pathogen product is ingested by the host, with the former being much more common than the latter. Most bacterial infections result from entry of a pathogen into the body. Entry can occur through a break in the skin into the underlying soft tissue or at the surface of a mucous membrane of the respiratory, gastrointestinal, or genitourinary tract. The skin may also be directly infected. Entry at these sites may result in infection of the bloodstream, which may in turn lead to infection of other organ systems. Entry into the respiratory tract occurs via respiratory droplet nuclei, airborne particles 1 to 5 micrometer in diameter, or via fomites. Fomites are objects or materials which are likely to carry infection, such as clothes, utensils, doorknobs, and furniture. Infectious droplet nuclei are generated when an individual with a communicable respiratory infection, example tuberculosis, Legionnaire's disease, psittacosis, influenza, measles, chickenpox, aspergillosis, COVID-19 infection, sneezes, coughs, talks, plays a musical instrument by mouth, or sings. A cough may generate 3,000 particles. A sneeze may generate up to 40,000 particles. Large particles may evaporate down to the 0.5 to 12 micrometer range, and particles that are 1.5 micrometer and hygroscopic increase in size as they pass through the moist nasal passages and lower respiratory tree. Entry into the gastrointestinal tract occurs via ingestion of contaminated food or water or via person-to-person contact, typically with transfer from a contaminated hand into the mouth. Pathogens for which the infectious inoculum is large. Example, 10 power 8 organisms for epidemic spread of Vibrio cholerae, 10 power 5 organisms for typhoid fever, are generally acquired via contaminated food or water, whereas pathogens for which the infectious inoculum is small, example 10 power 1 that is 10 to 10 power 2 that is 100 organisms for Shigella species are more commonly acquired by person-to-person spread. Entry into the genitourinary tract generally takes place either via colonization of the urethral meatus, that is the entry point into the urinary passages, or vaginal introitus, that is entry point into the vagina, with focal organisms, followed by ascension of the organisms into the bladder or kidneys, or via instrumentation. Pyelonephritis, that is kidney infection, can also result from seeding from the bloodstream. Establishment of infection. Preferred areas for organisms. Live pathogens. Within the human host, many bacterial pathogens display tissue tropism, that is affinity. The sites of infection within the human body are pathogen specific and restricted. Pathogens decide on which tissue they prefer, such that even adjacent tissues may be uninvolved. For example, group A streptococci cause pharyngitis, that is throat infection, and soft tissue infection in the skin, but rarely pneumonia, that is lung. Cholera is an infection of the small intestine, but not the stomach or the colon. Shigella species prefer the rectosigmoid, that is the lower part of colon. Therefore, to establish infection, the pathogen must access its niche, that is preferred area, and then remain within that niche. The following list shows the bacteria, their favorite sites of residence, and the diseases they cause. Disease, pertussis or whooping cough, caused by Bordetella pertussis. Favorite site, respiratory tract. Disease, chlamydia including trachoma, caused by Chlamydia trachomatis. Favorite sites, eye and venereal organs. Disease, colitis, that is inflammation of large intestine, caused by Clostridium difficile. Favorite sites, colon, large intestine. Disease, tuberculosis, caused by Mycobacterium tuberculosis. Favorite sites, respiratory tract. Gonorrhea, caused by Neisseria gonorrhoeae. Favorite site, urogenital tract. Typhoid fever, caused by Salmonella typhi, residing in the gastrointestinal tract. Favorite site. Disease, dysentery, caused by Shigella. Favorite site, colon and rectum. Disease, cholera, caused by Vibrio cholerae. Favorite site, small intestine. Disease, plague, Yersinia pestis is the causative organism and afflicting internal organs. So, dear viewers, this is the end of video part 4 in the 4-part series on infections overview in the Medicine for Anyone in the YouTube channel. Thank you for watching.